Hey guys, I'm Jasmine and I'm here to show you if it's possible to have a duplicate.
Have you ever wanted someone just like you? Someone who shares the same DNA as you? Or maybe you're just interested in cloning, but either way, science does say it's possible. Before we get into all the sciencey details, you may be wondering, what exactly is cloning? You may see it in movies or TV shows, but what exactly is cloning? So, to sum it all up, cloning is basically organisms that have the same exact genetics. Now, there are two different kinds of cloning that happens naturally or in the lab. Now, in the lab involves white coats, scientists, and petri dishes, but naturally is your everyday identical twins, and plants do it too. Um, whoa, not that kind of closer. But moving on. In the world of cloning, there are three kinds: gene, reproductive, and therapeutic cloning. Today, we're going to look at gene cloning. So, let's go. Gene cloning starts with a small segment of DNA called plasmid, which is extracted from the bacteria or yeast cell. This small circle are alongside the regular bacteria DNA. Plasmid is important because it helps the DNA resist deadly foreign substances. Now, in order to clone a gene, a small section is cut out of the plasmid with restriction enzymes that act as molecular scissors. The two ends of the circle are now called sticky ends. Then some of the DNA is extracted from the organism. This is called an isolated gene. In this case, we are using human insulin. Now take the human DNA that you wish to be cloned and the plasmid. Now the human insulin is inserted into the gap with another enzyme that will act like tape. This enzyme is called DNA elastase enzyme. And now we get our modified plasmid. Each plasmid carries a unique part of the foreign DNA. These are called pools or libraries. Now we take the plasmid and put it back into its host cell. This process is called transformation. Now the modified cells are put into petri dishes to grow. Plasmid uses their host cells' enzymes to replicate themselves, like this, thus creating more cells, which makes colonies also known as clones. So to basically wrap everything up, this process of gene cloning has been used on farm animals to make their bone structure stronger and to help them reduce the feeling of pain. And it also has been used in medication, but never directly on humans because plasmids carry functional genes throughout the human body, and this may lead to other health conditions and diseases to humans. And there would be a reduction of genetic diversity because we're being cloned. So it's pretty crazy, and that's all. Bye.